Good evening, class. Welcome back to microbiology. This is Silvana O'Coney, and in today's lecture we're going to take a look at prokaryotic reproduction and growth. First, we'll focus on binary fission, which is the type of cell division most frequently observed among the prokaryotes. Cell reproduction leads to an increase in population size, so we're going to consider population growth. Then we'll take a look at the influence of environmental factors on microbial growth. So we've seen that microorganisms must be able to respond to variations in nutrient levels and particularly to nutrient limitation. But the growth of microorganisms is also greatly affected by the chemical and physical nature of their surroundings. An understanding of environmental influences aids in the control of microbial growth and the study of the ecological distribution of microorganisms. Most microorganisms grow in fairly moderate environmental conditions, but then we have microbes that grow in extreme environments, and they're known as extremophiles. So now we're going to review how some of the most important environmental factors affect microbial growth. This table summarises the way in which microorganisms are categorised in terms of their response to different environmental factors. But it's important to note that for most environmental factors, a range of levels supports growth of a microbe. So for example, a microbe might exhibit optimum growth at pH 7, but will still grow, although not optimally, at pH values down to pH 6 and up to pH 8. In addition, outside this range, the microbe might cease to reproduce, but it might still remain viable for some time. So clearly each microorganism must possess adaptations that allows it to adjust its physiology within its preferred range, and it may also have adaptations that protect it in environments outside this range. So let's start by looking at solutes and water activity. Consider what happens if a microorganism is placed in a hypotonic solution, that is one with a lower osmotic concentration. Water will enter the cell and cause it to burst. Conversely, if the microorganism is placed in a hypertonic solution, that is one with a higher osmotic concentration, water will flow out of the cell. In microorganisms that have cell walls, the membrane will shrink away from the cell wall, a process called plasmolysis, and this dehydration of the cell in a hypertonic environment may damage the cell membrane and cause the cell to become metabolically inactive. So it's important then that microbes be able to respond to changes in the osmotic concentrations of their environment. And because this osmotic concentration has such profound effects on microorganisms, it's useful to be able to express the degree of water availability. Microbiologists generally use the term water activity for this purpose. Water availability may also be expressed as water potential, which is related to water activity. Water activity is reduced by interaction with solute molecules, termed the osmotic effect. So the higher the concentration of solutes, the lower the water activity. The water activity of a solution is 1/100th the relative humidity of the solution when expressed as a percent. It is also equivalent to the ratio of the solution's vapor pressure to that of pure water. Here we have the approximate lower water activity limits for microbial growth. And you can see that microorganisms differ considerably in their ability to adapt to habitats with low water activity. A microorganism needs to expend extra effort to grow in a habitat with a low water activity value because it must maintain a high internal solute concentration in order to retain water. Some microorganisms can do this and are termed osmotolerant. That is, they'll grow over wide ranges of water activity or osmotic concentration. So, for example, Staphylococcus aureus is said to be halotolerant and can be cultured in media containing sodium chloride concentration up to about 3 molar. So it's well adapted for growth on the skin. The yeast Saccharomyces eureusii, which you can see on this slide here, will grow in sugar solutions with water activity values as low as 0.6. The photosynthetic protist, also on this slide, Dunaliella, tolerates sodium chloride concentrations from 1.7 molar through to a saturated solution. So although a few microorganisms are truly osmotolerant, most only grow well at water activities around 0.98, the approximate water activity for seawater or higher. And this is why drying food or adding large quantities of salt and sugar is so effective in preventing food spoilage. But interesting to note, as this table shows, many fungi are osmotolerant and thus particularly important in the spoilage of salted or dried foods. Osmotolerant organisms are those able to grow over wide ranges of water activity or osmotic concentration. One example is Staphylococcus aureus. Now, many of these microbes, whether they're in hypo or hypertonic environments, keep their internal osmotic concentration above that of the external environment, so that their plasma membrane is always pressed firmly against their cell wall. And they do this by accumulating what are called compatible solutes to increase their internal osmotic concentration. Compatible solutes are those that don't interfere with growth and metabolism when they're present at high intracellular concentrations. Most microbes increase their internal osmotic concentration in a hypertonic environment through synthesizing choline, betaine, proline, glutamic acid, and other amino acids, and often they employ elevated levels of potassium ions. Photosynthetic protists and fungi, on the other hand, tend to use sucrose and polyols, for example, arabitol, glycerol, and mannitol, for the same purpose. Some microorganisms are incredibly adapted to extreme hypertonic environments. Halophiles grow optimally in the presence of sodium chloride or other salts at a concentration above about 0.2 molar. Extreme halophiles have adapted so completely to hypertonic saline conditions that they actually require high levels of sodium chloride to grow, concentrations between about 2 molar and saturation, which is about 6.2 molar. The archaean, called Halobacterium, can be isolated from the Dead Sea, which is a salt lake between Israel and Jordan, also from the Great Salt Lake in Utah, and other aquatic habitats with salt concentrations that approach saturation. Halobacterium and other extremely halophilic prokaryotes accumulate these huge quantities of potassium in order to remain hypertonic to their environment. Their internal potassium concentration may reach as much as 7 molar. In addition to this, their enzymes, ribosomes, and various proteins require high potassium levels able to remain stable and active. In addition to this, the plasma membrane and cell wall of Halobacterium are actually stabilised by high concentrations of sodium ion. And if that sodium concentration decreases too much, the wall and the plasma membrane start to break down. Let's turn our attention now to the effects of pH. It's not surprising that pH has a dramatic effect on microbial growth. Each species of microbe has a definite pH growth range and pH growth optimum. On this slide we have the pH scale, which is familiar to you, and examples of substances such as acids and bases with different pH values. To the right of screen we have some microbial examples placed at their pH growth optima. Acidophiles have their growth optimum between pH 0 and pH 5.5, neutrophiles between pH 5.5 and 8, and alkalophiles prefer the pH range of 8 to 11.5. Extreme alkalophiles have growth optima at pH 10 or even higher. In general, different microbial groups tend to have characteristic pH preferences. So, for example, most bacteria and protists are neutrophiles. Most fungi prefer more acidic surroundings, about pH 4 to 6. Photosynthetic protists also tend to favour slight acidic environments. Many of the archaea are acidophiles. So, for example, the Archaean sulfolobus is a common inhabitant of acidic hot springs. It tends to grow well around pH 1 to 3. There's an Archaean called Ferroplasma and others that can actually grow at pH 0 or very close to it. So let's think about how pH affects microorganisms. Well, the first thing to note is that a drastic change in cytoplasmic pH can harm microorganisms by disrupting the plasma membrane or by inhibiting the activity of enzymes and other proteins. Most prokaryotes will die if the internal pH drops below pH 5 to 5.5. Changes in the external pH might also alter the ionisation of nutrient molecules in the environment and thereby reducing their availability to the organism. So microorganisms respond to external pHs, external pH changes rather, using several mechanisms that have been proposed. First thing to note is that the plasma membrane is impermeable to protons. So neutrophiles appear to exchange potassium for protons using an antiport transport system. Extreme alkalophiles like Bacillus alkalophilus maintain their internal pH close to neutrality by exchanging internal sodium ions for external protons. And internal buffering may also contribute to pH homeostasis. However, if the external pH becomes too acidic, then other mechanisms come into play. When the pH drops below about 5.5 to 6, then Salmonella enterica, for example, and E. coli can synthesize an array of new proteins as part of what's been called their acidic tolerance response. A proton ATPase contributes to this protective response either by making more ATP or by pumping protons out of the cell. Then if the external pH decreases again to 4.5 or lower, then proteins such as acid shock proteins and heat shock proteins are synthesized, and these are proteins that prevent the acid denaturation of proteins, and also aid in the refolding of proteins that have been denatured. Now you can imagine that microorganisms often change the pH of their own habitat, and this is because they produce acidic or basic metabolic waste products depending on the species of microbe. So for example, fermentative microorganisms form organic acids from carbohydrates, whereas chemolithotrophs like Thiobacillus oxidize reduced sulfur compounds to sulfuric acid. And other microorganisms make their environment more alkaline by generating ammonia through the degradation of amino acids. So because microorganisms change the pH of their surroundings, we need to use buffers in media to prevent growth inhibition by large pH changes in the lab. And a commonly used buffer is phosphate. This is a good example of buffering by a weak acid and its conjugate base.